Summer, a PhD researcher at the University of East Anglia. I research DNA and its mysterious structures. DNA can be thought of as the language of life, as it's the genetic instructions on how we make all organisms on Earth, including bacteria, plants, animals, and even ourselves, humans. It's similar to how a recipe book contains instructions on how to make cookies, or how a Lego set contains instructions on how to build a castle. DNA is inherited from our biological parents, meaning that your mother and your father pass some of their DNA to you, which is why you can look similar to your parents and find physical similarities between you and them. Every individual has a unique DNA profile, and this allows us to identify criminals using DNA they've left at the scene of the crime. This could be from their hair, their sweat, their spit, or their blood. Imagine you are a forensic scientist at a crime scene. You enter a house that has been robbed. You find the broken window in the kitchen and see there's some blood on the shards of the glass. You swab the blood and take the blood sample back to the lab. The police have 3 suspects and have provided you with a sample of each of their DNA. You carry out a DNA profile analysis on all 3 suspects and the blood from the crime scene. Here are your results. Match the DNA profiles to find the robber. Suspect 3 is the robber, as their DNA matches that from the crime scene. DNA resembles a right-handed twisted ladder structure, which is known as the double helix. In DNA, the sides of the ladder are known as the backbone, which is made up of sugars and phosphates. And the steps of the ladder are made from DNA's 4-letter base code, where A stands for adenine, T for thymine, G for guanine, and C for cytosine. The DNA of all organisms on Earth is made up of these 4 bases and can be referred to as DNA's alphabet, making DNA similar to a language. We know that languages have rules. For example, in English, I comes before E except after C. Well, DNA also has base pairing rules, where A binds to T, as shown by the blue and red structure binding, and G binds to C, shown by the green and yellow structure binding. It's these pairing of these bases that form the steps on the ladder. However, just like in English, where in certain cases I does not come before E, these DNA bases don't always follow these base pairing rules, and it's that rule-breaking behaviour that allows DNA's mysterious other structures to form. One of these structures is known as the I motif, and it resembles a knot. The I motif forms because the rule where G normally binds to C, which was shown by green binding to yellow, is broken, and the C binds to another C, demonstrated by 2 yellow structures binding. Scientific research suggests that I motif structures are a way to turn on and off genes. Genes are sequences of DNA bases that instruct the body how to make proteins, in the way a recipe book tells us how to make a cake. Proteins are essential for normal and healthy cell function and do pretty much all of the jobs in our body, including making up our bones and our muscles, protecting us from viruses like COVID-19, and taking in nutrients from the food to convert it to energy. It's essential that we can control how much of each protein is made, because during different events, we need different amounts of different proteins. For example, I need more energy if I'm running compared to sitting down. So I need more of the proteins that can convert more energy so that I'm able to run. The I motif could be a vital method of controlling the protein production. They can also be involved in genetic diseases, which are diseases affected by our DNA, such as diabetes and cancer. I motifs could be the key to unravelling mysteries behind these diseases and could be powerful targets for new medicines. Therefore, I motifs could be used to save lives, and that's why they're important. I'm going to show you how you can make candy DNA models at home, and yes, you can eat them after. You will need some sweets that come in 4 different colours, like jelly babies. Each colour will represent a different DNA base. Some pencil sweets to represent the DNA backbone, and some toothpicks to represent the bonding that holds the structure together. Please be careful not to prick yourself with these. Firstly, separate your sweets into their 4 colours and assign each colour a base, like I have here. Then take a toothpick and attach your base pairs onto your toothpick, following your base pairing rules of A binding to T and G binding to C. Take your toothpick with your base pairs and attach it to your pencil backbone, like so. Then if you continue doing this until you have a full ladder like this one I made earlier, then you can pick up your DNA model and give it a clockwise twist to make a right-handed twisted ladder structure, like so. And there you have your candy DNA model. Don't forget to take a picture and share it with us on social media before you eat this delicious DNA treat. You can find a full list of instructions and examples in our downloadable pack on the link shown on the screen. We're also running a logo design competition on social media. The logo will need to use either the twisted ladder DNA structure or eye motif DNA, or both, and must fit Instagram's profile size recommendations. The winner's logo will be used as our Instagram profile picture, and for branding purposes, the winner will also be receiving a certificate. This competition closes on the 9th of November. For further details, please follow the link.
Hello, my name is Marco Cominetti. I work in the School of Pharmacy at the University of East Anglia. I'm a medicinal chemist, and my job is to research and develop new drugs. Today, I will tell you a bit about what I do, but also I will try to give you an idea of how very complex things can in fact be made up of a few simple building blocks. A bit like Lego, you can use the same building blocks to make an airplane or a car. And this applies to our body as well. Our body contains proteins, and all proteins are made up of only 20 different building blocks, and we call them amino acids. These amino acids can be made by our own body or obtained from food by disassembling the proteins which are present in it. If you think about it, it's pretty amazing that with only 20 amino acids, our body can make about 20,000 proteins. And they have very different shapes and very different roles. For example, they allow us to move because they make up our muscles, but they also send signals, and they allow our body to build things, amino acids, even proteins, and also something we call the DNA. The DNA is a sort of instruction book which allows our body to know how to make the proteins. And do you remember I just said about having about 20,000 different proteins in our body? Well, the DNA is made of only 4 building blocks. And this means that the instructions to build those 20,000 proteins are written with only 4 letters, which is pretty amazing. Now we have an instruction book. We have lots of proteins which do very different things, but how can they do all of these things? Well, the reason is because they can recognize and talk to each other. And this happens because they have very specific shapes and properties which allow them to recognize each other. It's a bit like a key and a keyhole. If they have the right shape, then they will allow you to open the door. It won't work with any shape, any key. And it's this recognition of 2 things, protein with DNA, that allow the proteins to do things, to trigger an event or prevent an event. We've prepared an activity pack. You can find the link with the video, and it will allow you to build some paper models. These represent proteins. We have a fixed one, which acts as a sort of keyhole. We'll call it a receptor. It has colors which represent the properties of it. And we will have to assemble the second one. We will call it a ligand. It represents another protein, so that the shape and the color of it will match those of the receptor, so that they will be able to recognize each other. But how does this tie into our work? Why is it important? Well, you may have heard of cancer. And cancer happens because the DNA, our instruction book, gets damaged. Usually it happens later in life because it's exactly like a book being used for a long time, it may start to wear out. And this means that some instructions may not be readable anymore, and the body will not be able to make some proteins or make them in the wrong way. And that in turn results in cancer. Nowadays we have treatments, we can even cure it, but we still need to get better at it. And what we try to do in the Cersei and Beekman groups at UEA is exactly to take advantage of this ability of proteins to recognize each other to make new drugs. In a specific case, we have two proteins which, by interacting with each other, protect the cancer. If we can prevent them from interacting with each other, then we can kill the cancer. And how do we do that? Well, we know the shape of one of them, so we build something different with the right shape and the right properties to match our receptor. And that means the original protein will not be able to bind anymore, and the cancer will die. It's, if you want, a bit like filling a keyhole with clay so that the key doesn't fit anymore. Just please don't try that at home. Obviously, I've made this very simple with a few paper models, but the reality is that a project like this requires a number of people with very different skills, which range from building computer models to doing chemistry, biology, analysis in the lab. And for this reason, we work as groups, and we also collaborate with other groups within the school and outside the school to do our research. The message to take home is teamwork gets things done. Then this is all for today. Thank you very much for listening. I hope you enjoyed it, and hopefully see you next time.